Okay, after chapter 3, you'll be glad to know chapter 4, we don't cover most of it. And so, really short amount of information here and just the basics. I'll tell you what to focus on. So we've already talked about metabolism is important, and it is all the chemical reactions found in your body. Cellular metabolism is the chemical reactions all occurring in the cell, and it's usually some pathway or some cycle to control it. So all cells perform types of reactions, catabolic and anabolic, which is like reactions where you're breaking complex structures down into simple, and anabolic is taking simple structures and building complex structures out of it. You won't be tested on that vocabulary, but if you were wondering what that meant. How fast that all happens has to be very controlled. If your cell were to release too much energy at once, first of all, it's never 100% efficient, so a lot of that energy will be lost as heat, and it could destroy the cell, which will eventually destroy the tissue, organ, and you would combust, right? So what the cell uses is what are called enzymes, and they control the rate and actually speed up chemical reactions. So if it were me, I would underline enzymes here, star this, and put speeds up chemical reactions, not just breaking things down or building them, but all reactions inside the cell. Now, enzymes are made of protein. They're globular proteins, and they have a very specific shape. That shouldn't surprise you because structure determines function. And so they catalyze specific reactions. And they do this by what's called lowering the activation energy to start the reaction. So if you were to graph energy, let's say, I don't know, you're taking sugar and you're going to break it down for the energy in the sugar into CO2 and water. So you would be up here with the amount of energy. This is like free energy or available energy. But to actually change sugar to CO2, it requires a little bit of energy called activation energy. And what happens is once you get over this hump, that rest of the energy in that sugar can be released, and you're left with these products. Remember the word reactants, yield products. So these products, CO2 and water, are no longer have as much energy in them, which is why we would never burn water or burn CO2. But sugar does burn, but you have to give it a little bit of energy. And then look at, let me change color here. So this amount of energy right here, we could measure, let's say it's like 1 unit of energy. That is the activation energy. The amount of energy released, notice while we had to give some energy, look at the amount of energy that's actually released from that process. It's quite a bit. So a little bit of energy is okay for the cell to give, to sacrifice, if you will, because the yield is so much higher. Now, when you have an enzyme, what happens, I'm going to choose black, is you start off with the same reactants and you're going to end with the same products. But what it does is creates a little microenvironment that lowers the amount of activation energy, and you get to that product faster. So this would be like time. And so it's the same reaction, just sped up. And how is it sped up? Because now look at the amount of energy that the activation energy, it's like half the amount of the activation energy required originally. And so that's how enzymes work. So when they talk about it lowers the activation energy, they're talking about this amount of energy needed to activate the reaction. And so how does it do this? Well, the enzyme has a specific place on it called the active site. That's here. You want to underline that or highlight that. And this is where a substrate will fit. And I'll show you pictures say 1,000 words. I do want you to know, if you can learn vocabulary, you can figure out almost any word on any test, even if you haven't learned the word. But if something ends with ACE, that usually means it is an enzyme. So lipase breaks down lipids. Sucrase breaks down sucrose. Lactase breaks down lactose. Nuclease breaks down nucleotides. And so you want to put that suffix in your pocket. People get that confused with this suffix, ose. If something ends in ose, it's a sugar. Glucose, lactose, sucrose, fructose, maltose. All of those, deoxyribose, that means sugar. ACE means enzyme. And a lot of ACEs, enzymes, break down a lot of oses, the sugars. So here you see the enzyme molecule made of protein has a very funky specific shape for these specific substrate molecules. So remember I said sometimes it's not breaking things down like my example said, the sugar being turned into CO2, but it might be anabolic reactions where we're actually building something. And so, and I'll give you an example of that in a second. So what's happening is these are the reactants. We call them substrates when they're associated with an enzyme. And these substrates fit perfectly like a puzzle in the active site. And once that happens, you have what's called the enzyme substrate complex, because it's more than one thing. That's why they call it a complex. And it's the enzymes with the substrates. I love this name. It makes sense to me. And what comes out of that is a product. And notice the enzyme hasn't changed a bit. So what could this enzyme do? It could take two more substrates. You could just make this a cycle, and it could continue making these products quickly from these reactants, faster than the cell could do without the enzyme. The metabolome is all the small molecules in the metabolism that control metabolism. And there's a database that store this. You probably heard of these as metabolites. And they estimate that we have about 2,500 of them, and only less than half have even been identified. And what do they do, and how do they control all the pathways and cycles that occur in the body? A lot of times the drugs we take are influencing our enzymes in our cells. So a metabolic pathway, if I think about a pathway at my grandma's house, it has all these little steps. So this is a series of enzymes that control the formation of a final product. So as those two substrates will make a product, but that product becomes another substrate for the second enzyme, and the pathway continues. Now it is rate limiting because you're dependent on the first enzyme to make the second enzyme's substrate, and so it's kind of like making a pizza. You can't put toppings on unless the dough's been spread out. And you can't cook the pizza until the toppings have been on. So you're limited on the speed by who's in front of you. And so same thing with these enzymes. And of course, we are going to control this with negative feedback. And so let's see how this works. So I talked about pizza, like making dough, rolling dough, putting on cheese, tomato sauce or whatever the crap that is, and toppings, and then, cook it, and we have a pizza. You're not going to have pizza if this dude is really slow or this chick called into work. So that's kind of how these enzymes work. So what this, this is the enzyme. The arrows are the enzymes. So we start with this first substrate, for example, right here, and it fits in the active site of enzyme A. And enzyme A turns that molecule into a product, which happens to be a substrate for enzyme B, which makes a product that ends up being a substrate for enzyme C, and so forth, until you end with the product. Now, what will happen is that the accumulation of this product will actually bind to one of the enzymes in this case the first one, where if this product is blocking this from happening, well then eventually enzyme B is going to run out of its substrate and this whole chain of events is going to stop. Well, why would the cell want to stop? Because you have enough of this product. Okay. It's kind of like this is the process of making money. And when you accumulate millions of dollars, you're probably not going to work real hard again. And so what happens? You stop making money because you have enough. Now, what happens after you spend some of that money and use it? You might need to what? Start working again. And so the same thing happens here. As the product starts to be consumed by the cell or used for whatever purposes, it's going to unblock enzyme A so that we can start this metabolic pathway again to make more of the product needed. There are some things that alters enzymes. There's cofactors, coenzymes, and those are molecules that either help the enzyme function and make its shape, specifically the active site shape, work for that metabolic pathway. You can put little X's by this. I'm not going to test you on cofactor or coenzyme, but denaturation is when you inactivate the enzyme, and you can do this to other proteins besides enzyme. And it's how do you do that? You change its shape. So denaturation changes the shape. You might have heard of a protein being denatured. That just means something caused its shape to change. And a number of things can do that. Heat, extreme heat. Extreme cold does not denature it. It takes energy to change the shape. So extreme heat, extreme basics or extreme basic fluids or acidic fluids. Very salty water can do this to some enzymes. And so that's how, like, when you study the digestive system, for example, there's amylase in your saliva, and it works to break down sugars, and it works best in about a pH of 7. So if you're constantly eating, I don't know, if you, for whatever reason, had a lot of acid in your mouth, your amylase wouldn't break down that sugar for you well. And then you have gastric amylase, or pancreatic actually amylase, and it's going to work in more basic environments. So those enzymes wouldn't work well unless they're in the correct environment. If they're not in an appropriate environment, they might be denatured and no longer work. Sometimes this can be irreversible and cause damage to the protein. You do this when you cook a burger. You know, it changes shape. Like you make the patty, pound it out perfect for the bun size, and then you go cook it and they're half the size. That's because you denatured the protein and changed the shape. And fun fact is PKU, or phenylketonuria, is due to a non-functioning or missing enzyme that blocks conversion of amino acid phenylalanine into the amino acid tyrosine. And so you end up with excess phenylalanine, and that can poison the brain. So people with PKU have a special diet where they are not getting this enzyme consumed this amino acid consumed as much. So I just wanted to review with you real quick about ATP. Remember, that's our energy molecule, and that's what the cell uses as a currency to do work. And it does have 3 parts. But the most important thing I want you to know is it's called ATP for adenosine triphosphate. There's 3 phosphate groups. We get the energy out of this molecule by breaking the bond between the second and third. And so what you're left with is, if you break ATP, 3 phosphates, if you break the third phosphate off, you end up with just 2 phosphates. And so it becomes ADP. So this is an ATP molecule, and here's those 3 phosphates. It's this bond right here that you want to break to harvest the energy out. And when you break that phosphate off, what do you have now? You have ADP. Well, your mitochondria is able to, with oxygen, take ADP and with food, use the energy to put that phosphate back on. And so you're able to regenerate this molecule of ATP as long as your mitochondria and your cells are working. So a lot of people want to talk about, like, when you eat food and carbohydrates like starches, bread, or what have you, and you eat it, you do break it down by a process called hydrolysis into these monosaccharides. We've heard about those, the building blocks for carbohydrates. And you can do a few things with those. You can break those sugars down further for energy with the mitochondria. You can store the sugar as glycogen. Remember that polymer, that polysaccharide glycogen that's stored in our muscles and in our liver. You could even use the building blocks of that monosaccharide, CHO, and add a nitrogen to it and make amino acids. And what they're not showing here is if you also consume too much of this, you can change it into fat and store it. And so this is showing that, remember I told you, when you eat food, you're going to use the carbohydrates first to harvest ATP from your mitochondria. Do not worry about this process. This is very biology and not in the scope of our course. But you can also use fat, and you can see, like, the butter on this toast can easily fit into this process the same way you used the sugar. But if you don't have carbs and you don't have fat, you can break down the proteins and get energy from those as well. So I just wanted to show you that. Don't forget about DNA, our polymer that holds our genetic material. Inside the DNA, there is coding sequences of nucleotides that determine all of these structural components of the cell. And really not anything on here to be tested, but I thought it was some people are very interested in genetics, and so I wanted to give this to you to read. But this is a monomer of what nucleic acids are made of, nucleotides, and we've seen this before in the last or in the second chapter, and you can put them together and build RNA, or put them together to build DNA. And what's amazing about DNA is here's those structures, and it makes this shape called the double helix. And the double helix wraps around these little proteins, and you have chromatin. Remember chromatin being stored in the nucleus, and you can't really see it. But when the cell needs to divide, it'll undergo mitosis. And the chromatin in prophase will actually condense into what's called chromosomes. This is a metaphase chromosome because they took this specific chromosome from a metaphase plate. And DNA is useful for so many things. It is crazy the impact in many areas of our life that DNA has allowed us, and the DNA technology has allowed us to enhance, whether it be through fertility, curing disease, catching criminals, paternity tests, even like looking at royal families and their genetics. There's just so much to be said about that. Don't forget that DNA can self-replicate, and that happens during S phase. So remember, we'll have our chromatin here. The chromatin will replicate here. The cell gets ready to divide, and in prophase, the chromatin turns to chromosomes, and then they line up in the middle, and then they back away from each other, the copies of the chromosomes, and then in telophase, we end up with 2 cells after cytokinesis. Y'all, that is the end of this chapter. So I kept it under 20 minutes. Just one lab on enzymes here and a practice for you, very short, and then you'll be ready to test. You can always go back to your practices and do them 3 times to get the highest grade, and then they'll help you for the test. All right, good luck.